friends, we are going to discuss on fermentative products, principally the amino acids and vitamins today. We will cover here briefly about what amino acids and vitamins are and their uses, and then see what exactly happens when we are about to produce these two in the industrial scale. We will begin our discussion with amino acids first. What are amino acids? These are basically the building blocks of proteins in all the cells and essentially constitutes of the carboxylic group at one side and an amino group on the other side. The R side chain specifies a typical amino acid, and there are about 500 known amino acids. They are also important as neurotransmitter, and they are involved in several of these biosynthesis and transport mechanisms. Now, amino acids, as far as industrial importance goes, we are all familiar with the two terms aspartame and monosodium glutamate. Aspartame is actually aspartyl phenylalanine methyl ester, and this is the artificial sweetener so commonly used by most of the diabetic patients. And on the other hand, we have the testing salt called as the monosodium glutamate. This is nothing but the glutamic acid, an amino acid. We will first see about glutamic acid. Now, this is the structure of glutamic acid, a very simple kind of amino acid. We will then move on to see what exactly happened which allowed the synthesis of these kinds of compounds in the industry. First, it was discovered by Carl Heinrich Riethausen in 1866, and then later by Kikune Ikeda actually commercialized this because he developed the crystalline salt of monosodium glutamate. Now, this discovery is very important as far as industry giving food is concerned, because in the Chinese preparations, most often the testing salt that we generally employ is nothing but monosodium glutamate, and this gave a boost of many industries which flourished for producing glutamic acid. Kikune Ikeda also gave us an idea, or rather patented exactly to develop industrially these salts of monosodium glutamate. And in Japan, there are widespread use of this, and the company by the name Ajinomoto also flourishes, and we can at times call this salt commonly as Ajinomoto. Now, if we look into the biosynthesis of this kinds of glutamic acid, we will see that mostly transamination reactions are involved. This transamination means the moving or removal of the amino group from one amino acid to the other, resulting in the formation of another amino acid. If we can see here, glutamic acid, this is our product which should be obtained. Now, during biosynthesis, α-ketoglutarate acid, which is a common intermediate of the TCA or the Krebs cycle, is transaminated to give rise to glutamic acid. We can see this transamination reaction in still more detail in this diagram. What happens? We can see here alanine with the amino group. This is donated to alpha-ketoglutarate acid, which is a keto acid. So, the amino group gets transferred, and this alpha-ketoglutarate gets converted to glutamic acid simply by incorporation of this amino group. While this alanine, the amino acid, on losing the amino group gets converted to another keto acid called as pyruvic acid. Now, these are brought about by transaminase enzymes. Now, coming to the organism which produces glutamic acid. There are many, but most importantly, the organism which has been strain developed for production of glutamic acid is Corynebacterium glutamicum. The name has been given on the basis that this organism produces the best yield of glutamic acid industrially. Now, these are gram-positive rod-shaped cells, and at times for amino acid productions, these have been converted to their auxotropic mutants. There are other organisms also involved, like the Brevibacterium linens and other species of Brevibacterium, which also gives us a huge amount of glutamic acid in the yield. Now, if we look into the production of glutamic acid, we will see that basically the sugarcane is used here as the carbohydrate source. The sugars are taken. Sometimes even molasses is importantly used here. After the molasses has been extracted, that is incorporated as medium into the fermentation tank and is inoculated by the required organism, that is Corynebacterium glutamicum in most cases. After this, we will see what happens during the fermentation stages, that is within the bioreactor, what are the changes which goes on. The cell now here will take up the nutrients. Now these are the sugar molecules which are taken up by the cells. Later the sugar molecules are increased in, or rather they go on taking up nutrition, and the cell, inside the cell we will see huge amount of the sugar. Now they will metabolize it and for their own need will produce glutamic acid. Now this we can see here in this cell, as the time progresses, the sugars are being converted, utilized, and synthesis of amino acid is taking place, and the green colored dots that we can see here representing the glutamic acids are being increased. So later there has to be mechanism which excretes out the glutamic acid from the cell. Otherwise, there will be a feedback inhibition, which means excess of glutamic acid within the cell will not allow the pathway to generate more of the amino acid. So once the acids are excreted out into the medium, we can see in the fermenter they will be in the broth, which needs to be harvested. So after that, once the broth is taken out and harvested, clarification steps or recovery steps will be taken, and that is mainly crystallization of monosodium glutamate here. Then drying is often preferred, which is then goes for packaging or bottling. Now basically there is a point to be noted in case of the excretion of glutamic acid, which is very important. A model here shows that often the level of biotin or something, an antibiotic like penicillin or lysozyme can be used to generally allow the cell wall and the membrane to be slightly leaky, so that the glutamate which is synthesized within the cell can be excreted out. Otherwise, as already mentioned, there will be feedback inhibition. Then we are moving on to see lysine. Because we have already understood that the importance of glutamic acid is too much in the food industry, where it is called as a tasting salt, we will now see about lysine and its uses, the second amino acid in question. Now this is a structure of lysine. We can see the carbon skeleton diagram here and the modular diagram here. And next, moving on to biosynthesis. Now here there is a slight difference with respect to biosynthesis. Several organisms will produce lysine by different kinds of pathways, and there are broadly 4 different types of pathways. First we have the dehydrogenase pathway, then we have acetylase pathway, we have succinylase pathway, and we also have aminotransferase pathway. Now the initial part is the same of the pathway, which begins with L-aspartate, which is converted to aspartyl phosphate. Then we have the conversion to aspartate semialdehyde. Then the semialdehyde can generate methionine, threonine and isoleucine, that is, these are other amino acids. But if we need to continue with the lysine pathway, then this aspartate semialdehyde will be converted to dihydrodipicolinate, which will get converted to tetrahydrodipicolinate. Now there are a lot of incorporations here, and after the formation of tetrahydrodipicolinate, then the pathways are different. In most cases we see that a succinylase pathway are followed by a lot of bacteria. And what happens here, let us see. After the tetradihydropicolinate, there is a formation of succinyl 2-amino ketopymylate, from which we get a compound diaminopymylate, which is the next converted to LL-diaminopymylate and further mesodiaminopymylate. These are then converted to lysine. Now this is not as simple as we see here, because most of the organisms do not have all the required enzymes for all the pathways, and essentially maybe at times only the mesoform is produced or a mixture of L and mesoform is produced, and we know we will be requiring only the L-lysine form. So basically the conversion steps at the last involves some kinds of bioconversion which may convert the racemic mixture into only the L-lysine forms. Now let us see the microorganism which is involved in the production of lysine. It is the same as that used for the glutamic acid production, but this is an auxotropic mutant. Corynebacterium glutamicum, an auxotropic mutant, maybe a homoserine auxotrop, can be used for the production of lysine. There are several others also like Brevibacterium, but this gives the best result. E. coli can also be used at times for production of lysine. These follow the diamino acid pymalic acid pathway, which we have already discussed. Now, in case of the production, there are certain things which have to be considered. Lysine production takes place in simple submerged processes which employs aeration as well as mixing, and the carbon sources incorporated include mainly molasses. Most of the time, sugarcane molasses gives good amount of biotin, and thus regulation can be achieved very well. Biotin, like glutamic acid production, is also important here, which regulates the permeability so that the amino acid can leak out into the medium and avoid the problem of feedback inhibition. The other factors which should be considered is that the nitrogen amount is to be very much regulated, and most often ammonium salts are in use. It also helps in the balance of pH, which maintained as neutral pH. The products like lysine and glutamic acid can be obtained, as already discussed, by adding some salts for crystallization, and later can be used as in the pure form. Now, lysine on the other hand at times can be obtained in a crude form without going into too much downstreaming with the purer steps, so that it can be used as an animal feed, and most often lysine is used in the crude form as the feed grade form. If lysine is required for medicinal purposes, then downstreaming is more elaborate and purer forms of lysine can be obtained for medical usages. With the amino acids that we have seen so far about glutamic acid and lysine, aspartate and others are also often produced by the industrial microorganisms. Now we are going to see the next topic, that is vitamins as products of fermentation. Now vitamins, I think all of us know, are very important for the growth of all kinds of cells, and the deficiency of a single variety of vitamin can cause lot of problems. Vitamins are required in very trace amounts or less amount, but that whatever requirement dosages are there should be present. Now, if a person eats different kinds of foods in proper amount, the vitamins are obtained from the different food sources. But that often does not happen, and we suffer from some or the other kinds of vitamin deficiencies, which may show as eruptions on the skin or problems with eyes or such. Now, we are going to discuss two different vitamin production today here, most importantly employed, like the vitamin B2, which is riboflavin, and vitamin B12 or cyanocobalamin. These two are produced industrially, while other vitamins can be produced by chemical synthesis as well. Now, if we see here, begin with vitamin B2. This is the structure of riboflavin or vitamin B2, and importantly, this has a very good role to play with other kinds of metabolism within a cell. Now, the sources of vitamin B2 include cereals, nuts, milks, eggs, vegetables, and lean meat. The organism which is often employed in the industries to produce vitamin B2 is Asbia gossypi, at times also Erythrothecium gossypi, earlier called by this name. Now we can see here, this is a fungi, the filamentous fungi, Asbia gossypi. Lot of strain development have undergone, and we do have now improved strains and stored in the culture collection centers, which can produce good amount of vitamin B2. If we look into the biosynthesis of vitamin B2, we will see that the guanosine 5-triphosphate and ribulose 5-phosphate, these two are required in the production of riboflavin. We will see the steps in slight detail. The guanosine 5-triphosphate is converted to diamino ribosyl amino pyrimidinone 5-phosphate. This compound is again converted to 2,5-diamino 6-ributyl amino 4-pyrimidinone 5-phosphate. Now, once these conversions happen, later we have the compound ribulose 5-phosphate adding to it, which generates the compound called as 5-amino ributyl amino pyrimidinone. Now, these compounds are all circulated, and we have the dimethyl ributyl lumazine produced, from which we get the riboflavin. This is the complete structure of riboflavin. The production of riboflavin incorporates a submerged fermenter, and it takes approximately 7 days for the completion of the product. Once the product is produced, which incorporates aeration and agitation, downstreaming is often done to give us a crystal of the riboflavin compound. Then we are seeing the vitamin B12 or cyanocobalamin. Now basically vitamin B12 has a complex structure which incorporates cobalt, and essentially in the production medium cobalt must be used. We can see the Coryn ring system here, which is attached to the 5,6-dimethyl benzimidazole structure. A ribonucleotide is also attached to give a complex structure of vitamin B12. Now, in the biosynthesis steps, we'll see that there are again, like lysine, several pathways possible depending upon the organism. We will see the one that is mostly followed by the industrial strain. From 5-amino levulinic acid, uroporphyrinogen 3 is first produced. S-adenosyl methionine is involved here to give result to pre-Coryn 2 structure, which results in cyrohydrochlorine structure. Then there are several step reactions to generate the cobalt cyrohydrochlorine, that is incorporation of cobalt. Then there are other stages and final structure is produced, which is vitamin B12. These other stages that I was just mentioning incorporates the use of cobalt pre-Coryn 2 structure, a cobalt pre-Coryn 3 structure to finally generate the cobalamine structure. Now you can see here, if we use Salmonella typhimurium, this follows a straight pathway to generate cobalamine, while Bacillus megaterium follows this kind of pathway from uroporphyrinogen 3 to the cyrohydrochlorine as discussed earlier to give us a cobalamine in this fashion. Now microbes which generally produces, as the two have already been mentioned, Salmonella and the earlier one, we also have Propionibacterium friedenrichi and the Streptomyces olivaceus, these involved in the production of the vitamin B12 or cyanocobalamin. In the production stages, we will see that incorporation of glucose is again important. Pure glucose needs to be fed, and the betaine sources should be employed well. Nitrogen sources may include ammonium or nitrogen related products, can often be used to give us a controlled development of the vitamin B12 yield. Now all this so far, aeration and agitation obviously should be employed because we are using Streptomyces olivaceus, which is an actinomycetes, or at times Propionibacterium, which is a bacterium. These have two phases of aerobic and anaerobic growth and generates the whole compound in different stages. Not until the last stage is the final product derived. An addition of the ribulose and other structures that we have just mentioned is very much important as precursor molecules in the medium. And the uses of vitamin B12 can be several. There can be injections or other intramuscular preparations, as well as in simple medicinal forms which needs to be supplemented at times if required. So pure products must be obtained. Now thus we have seen a range of products beginning from amino acids like the glutamic acid, then lysine, and followed by vitamins B2 and B12, which give us an idea that several of these important essential compounds are generated from microbial biosynthesis and produced by fermentation technology. This have helped us to gain better health and benefits of added taste when seen into the total biotechnology field of the industrial microbiology sphere.